Okay, so the heck are we looking at here? All right, this is an egg. This is a yes. There will be lab today, Hugo. The lab today is going to be super simple. I mean, actually, let's say for lab this week, since it's technically supposed to be your analysis lab, we're obviously not going to be doing that. You guys can, stay in the Zoom session if you'd like to, just listen to me talk about the kidneys, or I can show you some three-dimensional models of the kidney and the nephron. But otherwise, you don't have to attend lab tonight. It's really just going to be a review of the urinary system. So hopefully that answers your question, Hugo. But yeah, like there's no like that exercise in the lab shouldn't be on the lab practical or on the lab assessment. Okay. So I'll make sure that those kinds of questions won't be put on there. It'll be anything from the 18th exercise in the lab, or the last exercise in the lab, will be all focused on just anatomy of the kidneys. So, knowing each of the structures and parts of the kidney, that'll be what that's based around. Okay. Okay. So yeah, good question. Good question. All right. So for this figure right here, this is an ovum surrounded by a couple sperm cells, all right, or a few. In reality, this is not the case during a fertilization event. There are hundreds of thousands of these little guys all surrounding and trying to get into this egg to cause a fertilization event. In reality, fertilization events there's a lot that goes into them, and we'll address some of that a little bit later. But the phenomenon of birth and gestation is very intricate. And there's a lot of things that go into it, and definitely, kind of looking at it at a macroscopic scale, it can look like it's inexplicable, right? But there's, we have pretty much mapped out most of the molecular processes that determine how things develop, to the point where we can compare human development to other things that we're related to, so other kinds of vertebrates, things like alligators or birds or chimpanzees, all these different things that we are technically related to, we develop in a very similar fashion. So yeah, like the phenomenon of birth and development and growth and development is very interesting to study. So what do we use a reproductive system for, right? Well, first and foremost, we use it to produce sex cells, or things that we call gametes, right? So gametes are, in a nutshell, just sperm and egg cells, right? So a sperm cell, like this little guy that's here in orange, and then the egg cell, this large thing that's here in blue, okay? So we'll talk a little bit about the energetics involved in generating these things, but in general, male gametes are far more inexpensive, or far less expensive is probably the better way to phrase that, to produce and maintain than female gametes. So there's some really crazy statistics that I'll throw at you, that go into, or that we'll talk about when we address the energetics involved here. But just to kind of summarize that, male gametes are produced pretty much until we die, right? So you can have an 80-year-old man who is still producing viable sperm cells. And whether or not he's able to utilize those in the traditional sense of the word, or I guess in the biblical sense of the word, is another story entirely. But even people who get to that age, even so men who get to that age, still technically, if everything is functioning appropriately, are still producing viable sperm cells, right? But that's not the same story for females. Females tend to not have most of their viable gametes left by the time they're about 40 years old. And by the time a female reaches about 30 years old, they've lost 90% of the viable gametes that they have. And to boot, all of the gametes that a female will have are what she's born with. Once a female is born, she does not produce or generate any new gametes. Each female is born with only the gametes that they're going to have for the rest of their life, and that's it. So evolutionarily and philosophically, that's kind of a conundrum. Why is it that males are able to produce new viable gametes all the time throughout their entire lifespan. But females don't have that same luxury. There's a lot of things that go into that. And evolutionarily, one thing that we think could explain that has to do with the energetics that go into generating a female gamete versus a male gamete. So right here, this picture shows you just that there's a clear size discrepancy between these two kinds of gametes. There's a lot more that goes into producing a female gamete as opposed to a male gamete. Okay. The other purpose, fertilization, right? So fertilization is when two gametes fuse, right? So in the sense of the sperm and egg cell here, during a fertilization event, one sperm cell is able to penetrate the protein capsule that encloses this egg cell right here. Once that occurs, then there's a cell-wide depolarization event that prevents any other sperm cells from being able to penetrate that barrier and administer their own genetic information. So sperm cells act very similarly to like a virus, in which they inject their genetic information into the host or into the ovum here, and then they're done, right? And then the sperm cell eventually dies off. But that genetic information getting in here is the most important part, right? So our reproductive system both generates these sperm cells and egg cells and provides the ability for them to meet and cause a fertilization event, right? And if you are female, you're lucky enough to be able to have the machinery to be able to satisfy nourishing this new ovum or this new zygote, as we would call after a fertilization event. And so the uterus and the fallopian tubes provide all of the necessary nutrition and energetic requirements to provide that new zygote and then eventual embryo with the necessary requirements in order to survive. And then you produce another structure called the placenta that provides an ability to regulate waste control and nutrition control with the developing fetus as well. So there's a lot of really, really cool physiology that goes into each of our systems, right? And then importantly as well, each of our organs that make up our reproductive system are very important for producing sex hormones. And what's kind of interesting too is that between males and females, we produce a lot of the same hormones. We just have differences in levels of those things, right? So with males, obviously males are going to have far more testosterone or far higher levels of testosterone than a stereotypical phenotypic female, versus a stereotypical phenotypic female is going to have much higher levels of estrogen than a stereotypical phenotypic male. So there's a lot of gray area between those things, and those levels can vary. But both males and females have both kinds of sex hormones. So every male is going to have some amount of estrogen. There's just different levels of it. And so that can be, controlled by a lot of different things. Some of it is genetics. A lot of it is diet, and things like exercise, behavior, all these kinds of things. Neurochemistry can all affect levels of these hormones, right? Okay, so male and female reproductive genitalia, all right? So here in the male, we have the penis and the glans penis at the tip, and then we've got our testicles right here. So our right and left testicles, singular would be testis or testes. And then we have a set of tissue that connects to the testicle called the epididymis. And the epididymis is a connection of tubes that link up to a large vascular network inside of the testicle where all of the sperm cells are produced. So the epididymis will then transfer those sperm cells through the ductus, or sometimes called vas deferens, from the testicle back into the body. It will loop around the bladder and then pass into another gland that sits back here just behind the bladder that we call the seminal vesicle. So that we've got a right and left seminal vesicle here that produces the seminal fluid to convert just the sperm into semen. So that's the significance of that. The seminal vesicle, along with the prostate, provide a bunch of nourishment like extra sugar and lubrication. And more importantly, it helps neutralize the pH of the environment of the urethra, because urine has depending on diet, can have, a fairly alkaline pH and sometimes a fairly acidic pH. And so prostatic secretions will help lubricate and neutralize the pH of the urethra so that the sperm can survive when they're ejected through an ejaculation event, right? So that's our general anatomy here. And then over on the female here, all of these structures are going to be what we call homologous, or they are related to each other in terms of development, right? So they serve a similar function, but in a different sense of the word. So for example, the ovary right here. So the ovary is serially homologous, or you can kind of ignore the word serial, but it is homologous to the testicle because the ovary produces estrogen and progesterone, and it also produces follicle stimulating hormone. And the testicle here produces the male equivalent of those things, namely estrogen. I'm sorry, not estrogen, testosterone. So the ovary and the testicle are related to one another, right? And embryologically, when we're developing, they are the same thing. It's just that with the production of testosterone due to genetics that are present on a Y chromosome, that causes the budding. Oh gosh, what's the embryological term? It'll come to me here in a bit. But the developing sex organ, whether it's an ovary or a testicle, will respond to those hormones. And if it's testosterone, then that initial, my gosh, that's going to bug me that I can't think of an embryological word for it. But that structure that's going to transform to either a testicle or an ovary. See, I've been doing yard work since about 9 o'clock this morning, so I apologize if I'm a little loopy here. But then that structure will then descend into a part of the skin that makes up the perineum, or the space between, in this case, the scrotum and the anus. And so that extension of the perineum will form a little sac that we call the scrotum, and that's going to play an important role here in a little bit. And so then let's see. So then we've got our other homologous structure to things like the epididymis in the female here, which would be the uterine tube. So the uterine tube is very similar to the vas deferens, right, or the ductus deferens, right? So it connects the ovary to the uterus right here. Okay. So then the uterus is a little tiny extension that you can kind of relate to the urethra, or probably more close to the prostate or the prostatic structure right here. So the uterus is obviously going to expand and allow for a zygote to develop into a fetus in there. And then the vaginal canal is going to be obviously related to the duct of the glans penis here. And then the vaginal canal is linked to the vaginal canal is completely separate from the urethra, obviously. But then the clitoris, or the glans clitoris, is actually related to the glans penis. They are both the same structure in the sense that all of the nerve endings that supply sensory information to the glans penis are the exact same ones that occur within the glans clitoris of the vaginal vestibule or the vulva, right? So what's kind of interesting, though, and this is kind of my hot take on all of this, or controversial opinion, mammary tissue is technically not part of the reproductive system. And the reason being is because babies or newborns do not need mammary tissue. It obviously helps with longevity, of course, and it's definitely advised for breastfeeding newborns, for antibody transfer between mom and the newborn, and a whole host of other health benefits. But the woman, or the female here, does not need mammary tissue in order to produce viable gametes. It's not related. You can supply the body with hormones that are produced within the mammary tissue that will compensate for that if necessary. And oftentimes, even if mammary tissue, even if it doesn't produce enough of those hormones, you can still produce viable gametes without the addition of mammary tissue. So mammary tissue itself is technically not necessary or not needed for successful production of gametes. So depending on who you talk to, mammary tissue technically shouldn't be part of the reproductive system. It should be part of the epidermis, or the integumentary system, because the tissue there is related to that embryologically. But for the sake of our book and our test and questions that you'll be asked, we're going to associate mammary tissue with the reproductive system. But just know that medically and biologically, it's actually not part of the reproductive system. Okay, so just kind of recapping our cell cycle here, right? So we've got two major processes for cell division. We have one called mitosis and then one called meiosis, all right? Mitosis is where we have a cell divide in half to produce a clone of itself. Okay, so I'll write that in our meeting chat here. So by that we mean same genetic material. Okay, so mitosis, a cell splits in half to create two cells that are exact clones of one another, right? But meiosis is very similar to mitosis, but except we divide an additional time and we create 4 cells instead of 2 that are genetically different from one another. So meiosis is where we we go through 2 rounds of cell division. Producing 4 cells that are genetically different. So that's an important distinction to make, because in each of our reproductive physiology, male and female, our gametic cells undergo both mitosis and meiosis, just at different points. And usually the gametophores are basically the stem cells that are going to produce either an ovum or a sperm cell. Both start out and divide mitotically to create these things. But once the actual mature sex cell is being produced, that process then shifts to meiosis. So kind of just briefly going over this here, the first round of meiosis here, so first meiotic division, meiosis 1, is very, very similar to mitosis in that ultimately we're going to have our cell here that has all of the genetic information inside of it, all of our chromosomes, right, which are just solidified regions of DNA. Okay, each of our chromosomes is going to carry genetic information on it that make us us. So during this division event, we've got a couple of different phases right here. So we have something called prophase, metaphase, anaphase, and then telophase. Okay, so those are our 4 major phases, and then you can even add in the extra bit where the cytoplasm of the cells separates and evens out. That's called cytokinesis. But we'll just go with these 4 major parts here. So during the first part of this entire process, all the DNA condenses into this little shape of an X that we call a chromosome. And we have it, in this case, color-coded to show you that these are different sets of chromosomes, okay? But importantly here, when preparing for meiosis, that genetic information gets replicated, right? So we're gonna reproduce that. And then what occurs during prophase that doesn't actually occur during mitosis is we have a bit where these homologous chromosomes, or chromosomes that are the same size but just in different colors, they can have little segments that cross over and swap bits of genetic information. That's what we call a tetrad. And another word for that is something called a synapse, right? So it forms a tetrad, and that exact connection between those regions of homologous DNA are what we call a synapse. Okay, so this is where we have a lot of our genetic variability come from. So this is why we are not exact clones of our parents. We're a blend of our parents. We may look more like one parent over the other, but that just has to do with how much crossing over occurs during the meiosis part of division here. Okay, so we have in prophase 1, we have our homologous chromosomes that are going to orient themselves relative to one another, and then move to this kind of imaginary line that separates the cell in half that we call the metaphase plate. Now, the important thing to note here is note the orientation of these chromosomes across this metaphase plate. Okay, the entire chromosome in red right here, I'll zoom in. The entire chromosome in red is on one side of our cell, and the entire yellow chromosome is on its own side of the cell right here. So this is our line in the sand. Each of these chromosomes is completely on one side, right? So when we go through anaphase, next phase here, those chromosomes stay intact, but eventually move to opposite ends of the cell. Okay, so those chromosomes migrate to each side of the cell, and then we have two daughter cells here that are genetically different from one another, right? So our big red chromosome and big yellow chromosome are in separate cells. Small red chromosome, small yellow chromosome are in different cells, right? So this change or this division here is important, and it's an important distinction in comparison to mitosis, because mitosis is where we create exact copies of one another, right? But in meiosis, we are going to create genetically different cells from the parental cell. You can think of this cell that's up here at the beginning of the first meiotic division as the initial parental cell. And then all these 4 cells down here in the bottom right are going to be our 4 daughter cells. Okay, sorry. So then here's where it gets interesting. With meiosis, when we're producing our gametes, right? Remember, you get half of your genetic information from mom and half of your genetic information from dad, which is technically a lie, which we'll talk about in a sec here. But in essence, our sex cells have half of the genetic information that the rest of our somatic cells do. So your muscle cell has all 23 copies or 23 pairs of chromosomes in your body. But your sex cells only have 23 chromosomes in total. So normally we have 46 pairs in total. We have 46 individual chromosomes and 23 pairs. But in our sex cells, each of our sex cells only has 23 chromosomes in total, okay? It's got half of our genetic information. So each of these cells right here that went through, each of these 2 cells up here in part 6, these are the products of the first round of meiosis. So each of those cells is going to then divide again, right? So we go from each one of these cells, it's going to divide in half. So when we start out with 1 cell, we end up down here with 4 cells. But note here that during metaphase 2, or during the metaphase of meiosis 2, those chromosomes orient themselves across that metaphase plate. So these these chromosomes separate from one another, right? So ultimately, each of these cells that we end up with has half of the genetic information that they started with up here, right? So each of these chromosomes gets split in half, right? So this is why meiosis is what we call a reductionary division, whereas mitosis is a equatorial division. So any cell that undergoes mitosis is an exact clone of the parental cell. It ends up with the exact same amount of genetic information, the exact same genetic information that it started with. It's just replicated, okay? But with meiosis, we reduce the genetic information that's inside of each of these daughter cells by half, okay? So meiosis, 4 daughter cells that are genetically different from one another that have half as much genetic information, okay? So we'll circle back around to that. That's an important distinction here, and it's an important reason for variation within offspring. But for a test or something, you should definitely know each of these steps of meiosis and why it's different from mitosis. Okay, so here we've got a sagittal section through the male reproductive system, right? So this is right through the pubic symphysis, so right down the middle of the body here. And we can see a lot of things going on here, right? So we've got our urinary bladder up here. We have the coelom just above it. We can see the ureter connecting to it, coming down from the kidney. And then we have the sigmoid colon down here that's leading into the rectum, and then ultimately the anus down here. And so what we're seeing, all this stuff in red, this is all musculature that makes up the pelvic bowl or the perineal girdle that provides the basis for supporting all of our guts and our reproductive organs down here, right? So interesting things to know here. So let's talk about some of this stuff. So right over here, we obviously have the penis and then the glans penis with the prepuce or the foreskin, right? So we've got our cross section through here, and we'll talk a little bit more about this tissue in a bit. But just really quickly, we have two different kinds of tissue. One that's going to help prevent the urethra from being blocked off or occluded during an erection. And then we have the tissue that's just above that in purple here that provides, or that I should say manifests the erection, right? So this is the tissue that becomes engorged with blood during an erectile event, right? So if we look over here with the scrotum, so we have the scrotum down here. So you can see this epidermal layer that forms the scrotum right here. But what's interesting is inside the scrotum, we have 3 layers of musculature that all form right here. So we've got our cremaster muscle and dartus muscle down here, which are homologous to the abdominal muscles. So we've got 3 sets of abs. We've got our external obliques, our internal obliques, and then the transversus abdominis, the cremaster muscle and dartus muscle, and there's another muscle in here that are all related to those muscles because as the testicles descend during development, they pull that musculature with them into the scrotal sac right here, and then they transform into the musculature associated with elevating the testicles when it's cold, bringing them closer to the body when it's cold, or allowing them to relax when it's hot, and allowing the testicles to hang more freely within the scrotum, okay? And then we can see we've got the testicle itself here with the epididymis that's attached to it, and then the ductus deferens right here. So what's interesting is you can see the ductus deferens starts out outside of the body wall, but then enters the body cavity through an important hole that we'll mention a little bit later on called the inguinal canal, and that leads into the body cavity, comes up and around the pelvic bone, and then notice how it loops back around behind the ureter right here. Okay, so it loops back around by there and then down into the seminal vesicle. So the seminal vesicle is important because it produces seminal fluid, which is going to help nourish the sperm and provide them with nutrients to be able to, survive their journey. And then we've got the ejaculatory duct here. So as an ejaculation is occurring, the seminal vesicle will propel fluid through the ejaculatory duct into the urethra, the beginnings of the urethra right here. And right around here, we can see this is all the prostate gland. The prostate is also going to provide important secretions as well, as I had mentioned before, very necessary for neutralizing the pH of the urethra and also providing the now semen with important nutrients too. And then we have the urethra that's connected from the glans penis to the urinary bladder here. But then there's another gland right here called the bulbourethral gland. Sometimes it's called the Cowper's gland. Bulbourethral gland also produces secretions that are going to help lubricate the urethra. So it's the pre-ejaculatory fluid that is produced by this gland, this little tiny guy right here. Okay. Let's see if there's anything else I wanted to mention here. Okay. Anybody have questions or anything so far? We good? Thumbs up? You're good? Okay, cool. Okay. Also, if you guys have questions about things, today and Thursday, this is the time to throw them out there. If you guys are curious about any of those things, we'll have a little bit of time on Thursday and potentially today as well, to talk about some, like, sexual education, if that's what you guys are interested in. Please, there's no weird questions. Like, don't feel awkward for asking anything. This is anatomy. This is what we're here to learn about, right? And I will answer to the best of my ability. And if you guys feel comfortable asking them, then definitely throw those things out there. Okay. All right. So let's take a look at the microanatomy of the testicle here. So if we take a section through the testicle, we can see that there are individual septa that separate the testicle into different lobes. Okay. So each of these lobes here houses a collection of what we call seminal vesicles or seminiferous tubules. So these are going to house the cells that are going to be producing new sperm cells. All right. So they're going to produce or undergo what we call spermatogenesis. In the case of females, this is what's going to occur within the ovum. I'm sorry, not in the ovum, within the ovary, called oogenesis. All right. So these lobules with the coiled seminiferous tubules are where we're going to be producing new sperm cells here, which we'll talk about more in a sec. So all of these tubes are connected to a little vascular network that we call a rete. So this is the rete testis, which is all of the connection of each of these seminiferous tubules. So essentially, if you are a sperm, then all of the little sperm cells are produced within these lobules and then move out this way into the epididymis, right? The other way that I like to think about it that's kind of funny is, all of these little sperm cells that are getting produced in here are training to be fighter pilots, right? So inside the testicle, this is like flight school, right? So all of the little sperm cells that are produced in here, they're just all kind of basic cadets. They have not earned their wings. They don't know how to fly yet, right? So once all of those sperm cells are produced in here, they go to flight school. So they have to mature before they are produced or they're considered viable sperm cells. So once they're considered mature, then they pass through the rete testes into the epididymis. This is where the sperm get their wings, all right? So the sperm cells mature and become fully functional sperm cells within the epididymis, right? And then during an ejaculatory event, sperm move through the epididymis into the ductus deferens via peristaltic smooth muscle contractions that are going to not pull, but more force sperm cells through this vascular network right here. Okay. So if we take a cross section through one of those seminiferous tubules, we have several different kinds of cells here. These little cells called spermatogonia are basically the starter cells for sperm cells. These are like the basic of the basic. They are not functional sperm cells in any way, shape, or form, but these spermatogonia end up undergoing several rounds of mitosis and then ultimately meiosis to produce little functional sperm cells in here. Okay, so each sperm cell kind of goes through levels of maturity that we call primary and secondary spermatocytes. All right, so then the primary and secondary spermatocytes will end up going through more rounds of meiotic division more rounds of mitotic division until ultimately they undergo 2 rounds of meiosis to be able to produce a fully functional spermatid. Okay, so the spermatid then undergoes another action where it has the midpiece and the tail here that get attached to the little head right here. So the head of the sperm cell, the nucleus, contains all of the genetic information from us, right? Or half of the genetic information, I should say. And then the centriole connected to this tail, this midpiece here, houses a bunch of mitochondria that burn through a bunch of sugar in order to provide the energy for the swimming motion that we see that's characteristic of sperm cells. Then the acrosome here, this is an important capsule that contains a bunch of enzymes that are able to degrade parts of the cell membrane of the ovum that allow the sperm cell to penetrate in there and deposit its genetic information during a fertilization event, right? So these sustentacular cells here in these Leydig cells provide nourishment for the seminiferous tubules. And they also provide molecular instructions to trigger the spermatogonia to undergo mitosis and then meiosis. So let's talk about that. So that's this next figure right here. So this is all of that stuff that we just mentioned, but kind of broken down in some steps right here. So spermatogonia, right, or spermatogonium, single, singular, this germ cell is going to undergo mitosis, right? So we're going to clone different forms of the spermatogonial cell, right? So each one of them are going to be able to mature into a fully functional sperm cell. And what's kind of crazy is, again, if we think of it in terms of bookkeeping, if we start out with one spermatogonium, that one daughter cell that is produced via this clonal division through mitosis, right? If we just track one, remember this is going to occur with this other clonal cell right here as well. This daughter cell is then going to integrate into a sustentacular cell, which is going to trigger it to undergo meiosis. So this primary spermatocyte here will then undergo meiosis and divide itself into a secondary spermatocyte. So that number 46, remember, is the total number of chromosomes that are present within our sex cell. And then after our first round of meiosis, we are going to divide that genetic information in half, right? So 23 chromosomes here, 23 chromosomes here. But then the amount of genetic information stays the same in each of these other cells, but the quality or whichever chromosomes are present in them is completely random, right? Or I shouldn't say it's completely random. I should say the way that they orient themselves across the metaphysical plate, so whether or not we are passing on traits that we got from our mom or our dad, is completely variable here, right? So secondary spermatocytes are then going to complete meiosis through that secondary meiotic division event, and then end up producing the head of the sperm cell with the acrosome, right? And then the spermatids are going to be attached to the bodies of the sperm cells, so the midpiece and the tail, and then end up entering the lumen, or the inside of the seminiferous tubule, as a sperm cell, or what we would consider to be a sperm cell, right? So this entire process is what we call spermatogenesis, right? So in males, this is occurring all throughout our lifetime, right? And if sperm cells are not ejaculated, then they end up just senescing, and they're broken down and recycled, and then we produce more, okay? So you can, I don't know if this is what any of you think of, or if this is what you believe, but you do not need to ejaculate sperm cells on a regular basis in order to have them, to prevent like a backup or a buildup of them or anything like that. That's not the case. If they aren't utilized, and they're broken down and recycled and then just produced anew, right? However, there is research that shows that regular ejaculatory events can help stimulate the prostate and help prevent prostate cancer. So there is a trade-off there. So, yeah, I've got a little note here about the descent of the testes. So really, as I mentioned, these are related to the ovaries, and in terms of embryological development, they are literally ovaries that tried to run away, but then ended up getting caught by the scrotum. It's literally like what ends up happening. So with the advent of that Y chromosome, carrying the genes to produce a bunch more testosterone, the testicles will then undergo dissension, or so they will descend, right? Sometimes what can happen during development and during birth is we can have something called cryptorchidism, right? So what that means is sometimes the testicles don't descend, right? So the nice thing is that this is very easily corrected via surgery. And so typically if, a newborn male has testicles that don't descend within the first 4 months of life, then usually you want to have that surgically corrected, because a couple things can happen. The first one is sterility. So if the testicles remain inside of the body wall, the testicles are then kept at too high of a temperature, and then they aren't able to produce viable sperm. So all the sperm that are in each of those testicles will die, and all the cellular machinery that we just talked about here won't function properly. So sterility will occur there. And then in worst case scenarios, sometimes the testicles will end up generating cancer cells, right? So if you have living tissue in here that's not doing anything, it's not undergoing proper cellular division events, then that's usually a breeding ground for things like cancer. And so there's some research and some questions that are going into maybe that's a reason why ovarian cancer is so common in females that are in later stages of life, and usually why things like hysterectomy are so common, because the ovaries end up developing tumors at a significant rate. So it's definitely, it could be related to that, but it's almost definitely related to the way that that egg cells are produced during ovulation. So we'll talk about that more on Thursday. But then what's also interesting is, since the testicles descend into the scrotum from the body cavity, there's a physical connection between the scrotum and the body cavity. And so there's a hole right in the groin that we call the inguinal canal. And that inguinal canal has an actual physical connection from the scrotum into the body cavity. So what that means is, sometimes, with things like when you sneeze, or if you're exercising and you pressurize your abdomen, you create a pressure in there, sometimes up to 90 psi, depending on the exertion. And in some cases, you can have something called a hernia. And a hernia is where you have some amount of tissue in your body. Usually it's the intestines, some other kind of tube inside of your body that's able to protrude or try and exit the body wall. And you can have abdominal hernias, where parts of the intestines will actually push through the muscular wall of the obliques or the rectus abdominis. But sometimes, if the pressure builds up so much in the abdomen, you can actually force parts of your intestines from the abdomen into the scrotum, which I've been told is quite painful. It is correctable via surgery. Sometimes they can go back on their own, depending on how much has herniated into the scrotum. But it's definitely something that, if it's not addressed, if it doesn't correct on its own fairly quickly, or if it's not addressed in a reasonable amount of time, if the vascular supply to the intestines is cut off for a prolonged period, then that tissue will senesce and die off, and that's really, really bad, because you can develop sepsis and die from that. So definitely make sure that you get that corrected. So yeah, hernias can definitely go away. I know I'm just kind of reading this as I might have already answered your question here, Alex, but yeah, hernias, depending on the severity of them, can either go away on their own, or oftentimes will need to be surgically corrected. Right? Okay, yeah, no, I'm sorry. I literally just read your question. I went into it without even looking at the chat to see if that was what you had asked. Okay. So here we've got a frontal section through the male reproductive anatomy as well, all right? So we've got our testicle here with the epididymis that's attached here, so all of our sperm end up getting their wings right here. Then we've got the ductus deferens that's connected to that. And then we've got our penis right here. Let's see, what else do I want to talk about? Okay, so yeah, with this structure right here, the spermatic cord, all right? So spermatic cord is made up of 5 different kinds of vascular tissue. And the nice thing is, 4 out of the 5 of them all have the word testicular in them. So we have the testicular artery, testicular vein, testicular nerve, testicular lymph, and ductus deferens all make up the spermatic cord. Okay, so there's 5 different structures in there that make up the spermatic cord. And then we have connective tissue and muscle that ends up covering that. So we have external connective tissue and muscular tissue that end up covering that. Okay. So the cremaster muscle covers that. All right. But those 5 things all make up the spermatic cord. The spermatic cord is the vascular network that connects the testicles to blood supply, because it's living tissue, lymphatic supply, as well as its own vascular network in order to do the job that it needs to do. All right. So then here, the spermatic cord, if we focus on just the vas deferens here for a second, we picture ourselves as a sperm cell undergoing an ejaculatory event. We start out in the testicle and we move into the epididymis. From there, through peristalsis, we move up the vas deferens right here, around behind the bladder, and then into the seminal vesicle right here. So this ampulla of the ductus deferens is literally just an expansion of that tube as it connects to the seminal vesicle. The seminal vesicle will then secrete nutrients and extra fluid into the mixture of sperm cells, converting the sperm cells into semen at that point. So one question that I could ask you on the lecture assessment for the final is, I could point to ductus deferens here and ask you if the cells in here are considered sperm or semen. And at this point here, we refer to them as semen because we haven't added any seminal fluid there. Okay? So the seminal fluid is only added once the sperm cells reach the seminal vesicle. So as we enter the ejaculatory duct here, now we're considered semen. All right. So then we enter the ejaculatory duct. We've got our bulbourethral gland, which is producing pre-ejaculatory fluid that's going to help lubricate the inside of the urethra. And then we have prostatic secretions here produced by the prostate gland that are going to help not only nourish the now semen here, but also help neutralize the pH environment of the urethra. All right. So let's see. So yeah, prostatic fluid, definitely very important for that. Another, quick tell by looking at the urinary system is that only males have a prostate gland. Females do not have a prostate gland. Only males do. Okay. So then, during an ejaculatory event, semen enters the ejaculatory duct, and then through peristaltic rhythmic contractions, they are expelled through the urethra into the unknown. Okay. So with the events of ejaculation, there's a lot of things that go into it. So we've got, emission, ejaculation, and then resolution, right? So the initial beginning of it starts off with extra hormones that are all controlled by the nervous system. Then we also have nitrous oxide that's produced that gets pumped into the what we call the corpora cavernosa, which is the erectile tissue that surrounds the penis, which helps engorge the blood vessels there, but it also prevents backflow of that blood into the systemic circuit. So during an erectile event, blood is shifted from the systemic circuit into the penis and it's kept there. So all of this tissue is literally like a sponge. It's a really rich network of capillaries that become engorged during an erectile event, and all that blood stays there. It's not allowed to return to the systemic circuit because we have sphincters that close off the blood flow of those blood vessels as the blood would normally try and exit the urethra right here. And so nitrous oxide is significant in controlling the return of blood into the systemic circuit. And then each of the parts of our autonomic nervous system kind of control different parts of the ejaculatory event. The parasympathetic nervous system is, used for the pointing action, and then the sympathetic nervous system is involved in the shooting action or the ejaculatory event. And so it's going to control all the smooth muscle contractions that cause the ejaculatory event. Okay, so here we have a cross section through the penis. So as we can see, there are two sets of corpora cavernosa right here. So one on the left and one on the right hand side. And then on the ventral side, we have another layer of tissue that surrounds the urethra called the corpus spongiosum. The corpus spongiosum is important because as the corpora cavernosa becomes engorged with blood, normally this tube, the urethra, would end up getting closed off, right? And that's going to kind of defeat the purpose. So the corpus spongiosum resists the corpora cavernosum and helps keep the urethra open during an erectile event, right? And then we've got the dorsal vein, dorsal arteries, and dorsal nerve right here that are on the dorsal side of the penis during the ejaculatory event, or during the erectile event. Okay, so hormones and how they're produced, especially through things like puberty. So puberty can happen, at various stages. Different people go through it at different times. The average age is around like 13 to 15 for most males. Some can, experience it younger. Some experience it a little bit later. It just kind of depends. But average age is around 13 to 15. So we have an important hormone called gonadotropin-releasing hormone, or GnRH, that is produced within the hypothalamus that stimulates the secretion of something called luteinizing hormone, or LH, and follicle-stimulating hormone from the anterior pituitary. So both LH and FSH are produced in males and females and act on the gonads, the testicles or the ovaries, and are going to be responsible for producing their target hormones, like in the male's case, testosterone here. So LH stimulates testosterone secretions from the interstitial cells of the testicles, and follicle-stimulating hormone stimulates the sustentacular cells of the seminiferous tubules to increase spermatogenesis and to secrete inhibin. So inhibin has a inhibitory effect on the production of luteinizing and follicle stimulating hormone. Okay, so testosterone has a stimulatory effect on the sustentacular cells of the seminiferous tubules, as well as on the development of reproductive organs and secondary sexual characteristics. Secondary sexual characteristics would be things like male features or, male features. So denser bones, more prominent brow ridge, sharper jawline, Adam's apple, or Joey's apple if you guys watch Friends or anything like that. Bigger muscle cross-sectional area. You can also have personality differences as well. You can have usually things that are associated with like elevated testosterone are things like confrontational attitude, so wanting to kind of enter conflict on a regular basis in order to solve issues, whether that's physical, verbal, anything like that. Is usually associated with elevated levels of testosterone. Usually, sometimes if they're in very, very high levels, they can lead to things like uncontrolled anger and rage. And a lot of the reason why teenagers tend to experience this quite significantly is because their bodies are producing very, very high levels of testosterone, because their luteinizing hormone is at an all-time high because they're undergoing puberty. Their body is preparing themselves to be ready for sexual reproduction. Okay, so testosterone has a stimulatory effect on sustentacular cells of the seminiferous tubules. They're going to be producing more sperm and extra secondary sexual characteristics. Testosterone also has a negative feedback effect on the hypothalamus and pituitary, and will thereby reduce gonadotropin-releasing hormone, luteinizing hormone, and follicle-stimulating hormone secretion. Inhibin has a negative feedback effect on the anterior pituitary to reduce follicle-stimulating hormone. So what's kind of interesting is testosterone, as it's produced, will inhibit itself, or will inhibit the overproduction of this, right? So sometimes these things don't necessarily or aren't inhibited the way that they should. And some things like male-induced color blindness can be a result of overproduction or overstimulation of testosterone secretion. One of my friends that I used to work out with, he was a ex-MMA fighter, and like the dude, you look at him and he looks like kind of a built guy, but he doesn't really look like he could squat over 500 pounds. But the guy had just like the thighs of tree trunks, and yeah, he was like, when I was exercising with him, he would tell me that like his testosterone levels were off the chart and he was colorblind because of it. So one of the reasons is that extra testosterone can actually fry the retinas within the eye and hinder color differentiation there. Okay, so really quickly, we'll kind of round out today with a discussion on hormones and kind of how they can act on the body. So GnRH, produced by the hypothalamus, target tissue anterior pituitary, which triggers the secretion of luteinizing and follicle-stimulating hormone. So luteinizing hormone stimulates synthesis and secretion of testosterone. Follicle-stimulating hormone supports spermatogenesis and inhibits secretion, right? So both of these are going to act on the testicles. Testosterone acts, in a ton of different places. So the development and maintenance of reproductive organs, spermatogenesis, causes the development and maintenance of secondary sexual characteristics, and inhibits GnRH, LH, and FSH through negative feedback. And then inhibin is produced or targets the anterior pituitary and inhibits follicle-stimulating hormone through negative feedback. So when everything's functioning properly, our body can control the amount of these secretions as they're produced and as they're needed. Okay. So that's pretty much it for male reproductive sex hormones. So luteinizing hormone, follicle-stimulating hormone, and testosterone, along with GnRH and inhibin. Okay. So then over here we've got the list of female reproductive hormones. So it's not a ton, but the effects of them are much more pronounced than they are in males. So GnRH, LH, and follicle-stimulating hormone all are produced as well here. But then we've got things like estrogen, progesterone, oxytocin, and human chorionic gonadotropin that are all produced in females that aren't produced in males. So let's see the effects of LH and FSH. So luteinizing hormone causes follicles to complete maturation and undergo ovulation, causes ovulation, and causes the ovulated follicle to become corpus luteum. So we'll talk about all of these structures more on Thursday. But each of these hormones, they're going to, where we have the production and synthesis of testosterone, supports spermatogenesis and inhibits secretion, we're going to have very similar effects in very similar structures. So if you think of the ovaries and the testicles as being related, the fallopian tubes or the uterine tubes and the vas deferens being related, the ampulla of the vas deferens and the fimbriae or the ampulla of the uterine tube, all of these things are going to be related, but the hormones in the female here are going to act in a little bit more of a specific fashion. So then we have follicle stimulating hormone that causes follicles to begin development. Okay, so follicles on the ovary. We have estrogen here, proliferation of endometrial cells, and also development of mammary glands, positive feedback before ovulation, resulting in increased luteinizing and follicle stimulating hormones. So kind of the opposite of the male condition. We have, instead of a negative feedback loop, we've got a positive feedback loop of the female sex hormone right here. Negative feedback with progesterone on the hypothalamus and anterior pituitary after ovulation, resulting in decreased luteinizing and follicle stimulating hormone secretion. So we have positive feedback, but then we can work against production of progesterone, which increases the production, or I'm sorry, decreases the production of luteinizing and follicle stimulating hormone. And it also acts on other tissues as well, development and maintenance of secondary female sexual characteristics. Okay? Progesterone, produced by the corpus luteum of the ovaries, is going to act on the uterus. So it's going to enlarge endometrial cells, and secretion of fluid from uterine glands helps maintain the pregnant state. Okay. A lot of birth control is going to affect and target things like progesterone and estrogen here to control the body and make it think that it's not, or that it's not pregnant and that it doesn't need to ovulate, right? Which is why a lot of the time during, or women who take birth control don't really experience a menstrual cycle, because those hormones are kind of under lower levels, and so the body doesn't think that it needs to ovulate, doesn't need to actually produce or shed its uterine lining or anything like that. Development of mammary glands, you got negative feedback loop, boom. Oxytocin, contraction of uterine smooth muscle and contraction of cells in the breast, resulting in milk letdown in lactating women. This is also the bonding hormone. So after birth, women they produce a lot more oxytocin in their brain when there's skin-to-skin contact with their newborn, and it helps with the bonding between the mother and the child. And then chorionic gonadotropin maintains corpus luteum and increases its its rate of progesterone secretion during the first third of pregnancy or the first trimester, and increases testosterone production in the testes of male fetuses. Okay, so some more effects of testosterone here, and then this is where we will end for today. So effects of testosterone on the penis and scrotum, enlargement and differentiation. So more testosterone, larger structures. Hair follicles, hair growth and coarser hair in the pubic area, legs, chest, axillary region, face, and occasionally back. Male pattern baldness on the head of the person has the appropriate genetic makeup. So male pattern baldness is actually linked to the maternal side of our genetic history. So chances are, if your mom's dad or anything like that, or if anybody on your mom's side of your family is bald, chances are you probably have the genetic makeup for male pattern baldness. My uncle and my grandpa on my mom's side are both bald. I'm doing okay, I think, so we'll see. By the time I'm 30 and then 40, if I've still got it, then I'm probably pretty good. Larynx, enlargement in larynx, deeper masculine voice, a deeper, more baritone voice allows for the expansion of those vocal cords, increases metabolism as well. Red blood cells, increased rate of red blood cell production. Red blood cell or hematocrit count is increased by 20% as a result of increased erythropoietin secretion. Retention of sodium and water to a small degree, resulting in increased extracellular volume. So cells tend to swell up a little bit in response to that. Skeletal muscle mass increased. Average increase is greater than that of females. rapid bone growth resulting in increased rate of growth, and early cessation of bone growth. Males who mature sexually at later age do not exhibit a rapid period of growth, but they grow for a longer time and become taller than men who mature earlier. Okay. So, this is why taking things like synthetic hormones can actually, anabolic steroids, for example, can affect a lot of this stuff. Part of the problem with hormones is that while they mimic and are actually, they affect things like muscle mass and bone development a lot more significantly than their natural biological counterparts, they have a negative feedback loop on things like gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone. So what that means is if you are supplementing your body with things like anabolic steroids, then your body says, okay, well, I don't need to have these organs that produce them on their own because I'm getting a lot more from an external source. So the organs that produce those things will begin to senesce or they'll die. So men who tend to take anabolic steroids for prolonged periods of times will experience testicular shrinking. So their testicles will actually end up shrinking and senescing, and sometimes they end up shriveling up and becoming non-existent. Okay. So that on that wonderful note is where we are going to end our discussion today. All right. Do you guys have any questions about anything? I've got the chat open here.